My name is Emma Rieser, and my project is called The Synergistic Effect of Caffeine and Hypocretin on Heart Rate and Sleeping Behavior in Danio. Located in the hypothalamus, selective cells secrete the neurotransmitter hypocretin. When hypocretin is activated, arousal in the body is sustained for a duration of time. Nonetheless, the absence of hypocretin's full threshold, as shown in the visual to the right, the activity promoted by the binding process becomes inconsistent. Frequent daytime drowsiness and interrupted sleeping schedules become side effects, along with the diagnosis for narcolepsy. Caffeine activates daytime performance. However, similar to narcolepsy, interrupted sleeping schedules become frequent due to a term referred to as caffeine withdrawal. Previous research demonstrated that high intakes of caffeine presented a greater likelihood to be tired the following morning than those who moderated their caffeine consumption to a minimum. The common median between these two is the animal model zebrafish. Like humans, the heart rate of zebrafish decreases from its normal 200 beats per minute to an average of 110 beats per minute. Additionally, the vertebrate stage of zebrafish, the hypothalamus contains 20 to 60 hypocretin neuron and receptors. Previous research showed that when exposed to a caffeine supplement, there was an almost immediate decrease in heart rate, then followed by a period of relatively stable heart rate, which ranged up to 170 minutes in zebrafish. Therefore, I hypothesized that if raising the concentration of hypocretin in Danio by consumption exposure, which leads to an increased heart rate, then caffeine's immediate effect when exposed to zebrafish is to decrease the heart rate, while simultaneously blocking the receptors of hypocretin. This is because following caffeine exposure in zebrafish, the levels of hypocretin will normalize, therefore opposing the categorizations of a narcolepsy progression. The research project was suited with 3 testing groups: the control group, the experimental group with hypocretin, and the experimental group with hypocretin and caffeine. The project began at 3 days post-fertilization. This was the age of the zebrafish. The experimental group's treatment involved administering 20 micrograms of Bacchum orexin, which is another name for hypocretin, into each of the wells present in rows B and C. 3 milliliters was distributed over a 3-day period. Here, consumption was used as a means to monitor diet through offering it in the aquarium water. Differing from the experimental portion of the research project, the control group in row A universally remained absent of scientific testing. Next, both groups were 6 days post-fertilization and observed under an inverted microscope programmed with OMAX camera software on a desktop computer. Through this, a 2-minute video was captured of each embryo and saved to the MacBook application QuickTime Player. This converted each video into slow motion, allowing for individual heartbeats of the zebrafish to be counted manually, utilizing a tally clicker. At 8 days post-fertilization, the experimental group with hypocretin and caffeine was carefully transferred into row D, where 0.75 milligrams of powdered caffeine was individually administered into each well. Each zebrafish was exposed to the caffeine-consuming environment for 60 minutes. This time interval was confirmed from past research to draw effects from the stimulant, but not cause fatality to the zebrafish. Utilizing the procedure from the heart rate data analysis slide, the heart rate of the zebrafish were recorded for 10 minutes, 30 minutes, and 60 minutes of exposure to the caffeine supplement. Figure 1 showcases the heart rate of the control group, experimental group with hypocretin, and the experimental group with hypocretin and caffeine after 6 days post-fertilization. Both experimental groups were exposed to a 20 microgram dosage of Bacchum orexin, which is another name for hypocretin, for 3 days. The hypothesis reflects the changes displayed on Figure 1, where it successfully predicts the subjection to hypocretin will increase the heart rate of both experimental groups. This is further emphasized when an ANOVA test was applied to the data as a whole, where a p-value less than 0.5 was revealed. The error bars on the graph indicates this farther. Therefore, Figure 1 serves as an accuracy test, demonstrating that hypocretin levels increased. Figure 2 illustrates the experimental group with hypocretin and caffeine's heart rate before exposure to the 0.75 milliliter dosage, during which and after. The hypothesis aligns with the results shown, showcasing that the caffeine exposure will decrease the heartbeat upon initial exposure. This is supported by the average heartbeat being 166.1 beats per minute before transferring the 6 zebrafish into the corresponding wells in row D. Then 30 minutes into the monitoring technique, the heart rate was recorded at 129.83 beats per minute. The decrease during exposure to caffeine mirrors that of the average zebrafish rate displayed by the control group in Figure 1 at 134 beats per minute. Figure 1 replicates previous research relating back to the broader picture of narcolepsy. A higher frequency of an increased heart rate in each subject was observed, demonstrating a relationship between the brain's hypocretin concentration and resulting bodily function. Figure 2 displays the effects of caffeine withdrawal. Upon exposure, the heart rate decreased to an average of 130 beats per minute for each of the 6 zebrafish compared to its initial pace of 166 beats per minute. The difference of 36 beats creates a correlation, whereas the slowed heart rate following caffeine intake was due to the stimulant's ability to block the receptors of hypocretin instead. An assumption can be made that the activity of the caffeine mimics that of a hypocretin deficiency in the body of zebrafish. The results produced through the experimentation contribute to closing the gap of caffeine's consequential effect on the body. Figure 2 can affirm that caffeine does promote a limiting effect to the body's function, shown through the decrease in heart rate demonstrating a high and the following increase resulting in the previous low. A warning created here is showcased through the known fact that the lower levels of neurotransmitter hypocretin is connected to narcolepsy diagnosis, the same of which occurs following the consumption of caffeine, though more gradually. Therefore, the broader picture can create a warning where hypocretin lost by the cup of coffee can lead to the development of issues larger than a couple hours of sleep lost. This current research project is under strict time constraints. Extending the time where both experimental groups were exposed to hypocretin would allow for a more confident analysis of preceding consequences. This also applies to the duration for which the diluted caffeine was exposed to the zebrafish of the single experimental group. And here are the picture references and the actual references. Thank you.